Are we up and running? Yeah. Well, I think we'll get started shortly. I welcome you to this program on the topic of Parkinson's disease. Two topics that will be talked about tonight have to do with the drugs and the things beyond drugs, and we'll also be holding a meeting tomorrow for our patient community to talk about similar matters. I'm Peter LeWitt. I'm here at Henry Ford Hospital, West Bloomfield, in the movement disorder and Parkinson's disease section, and I'll be introducing our guest speaker shortly. But before we do that, I'd like to acknowledge the support for this meeting of several of the vendors who are located in the hallway as you came in here in our Main Street district, and we thank them for their support and their interest in Parkinson's disease. These include Abbott DBS, Acadia Pharmaceuticals, Adamus Pharmaceuticals, Boston Scientific, GE Healthcare, Medtronic, and Pharmacy Advantage I see here, Residential Home Health, Rocksteady Boxing, and US World Meds. Don't want to leave them out. So we have a lot of support for this, as well as other services in the community. We thank you for your interest in our patient group, and we will proceed with the meeting. So as you can see up here, Giselle Petzinger is our first speaker. Dr. Petzinger is an associate professor and clinical scholar in the Department of Neurology at the University of Southern California, Keck School of Medicine. She completed her MD at USC, her residency in neurology at Yale University, and subsequently a movement disorders fellowship training at Columbia University. Her research investigates the topics of what you're going to hear today, and I think you're going to hear an exciting story of how to integrate the neuroscience of what's going on with what we all know. Exercise is good for you, but it's more than good for you. It's good for your brain. And she is currently supported by the National Parkinson's Disease Foundation, the Don Roberto Gonzalez Foundation, the NIH, and U.S. Army Department of Defense. And without further ado, I'd like to welcome her to our community and look forward to a very exciting talk.
Great. Thank you, Peter. Thank you. Well, I'm happy to be here, and I know I've got a lot of great colleagues in the audience who could also come up here and talk about their experience with people with Parkinson's and the whole exercise field, which is such a great community in and of itself. It's always wonderful to hear people's experiences and how they're helping. And so what I'm going to try to touch upon today, as Peter mentioned, I'm obviously not here to tell you that exercise is good, you know that. I'm going to tell you a little bit of a story about how we began to study it in the brain, and more specifically the different models we've used to understand what's happening in the brain in terms of plasticity. So with no further ado, let's go ahead and start introducing the topic. The first thing I always like to cover is some of the fundamentals of Parkinson's disease, and that becomes important because we're going to get back to the topic of plasticity. And one of the challenges we faced really early, so I've been in this field for a while, but we actually had a lot of criticism, a lot of pushback on the idea that how is plasticity relevant to a neurodegenerative disease, right? So we're talking Parkinson's, fundamentally we think of it as a cell loss problem, don't we? Right, and so this is a kind of traditional picture that we show, and what we're showing here is the area of the brain most affected. There are other areas, but the area of the brain most affected is what? The substantia nigra pars compacta, which is cell loss, and that's what we call a neurodegenerative disorder, a disorder of cell loss, right? But plasticity isn't about cell loss per se, or repairing or getting cells to come back, which we would call neurogenesis. What I'm going to be talking about here is slightly different. I'm going to be talking about circuitry, and this is where plasticity plays an important role, because plasticity is all about circuitry, you see. So I have to model this disorder in a way that makes sense in the context of plasticity. So now I'm going to draw you another picture. And this picture starts making a little bit more sense. It makes more sense for a lot of different reasons. One, I'm showing circuitry now, aren't I? I'm not showing cells, I'm showing circuitry. And the point is that the dopamine cells that make dopamine are actually modulating these circuits. All right, they're not the only neurotransmitter, there's other ones, but it's a modulator. Hence, to point out, dopamine in its system is not a behavioral circuit. It's working on behavioral circuits. These are behavioral circuits. The dopamine system is not a behavioral circuit, it's a modulator. It's interacting with these circuits. And what I'm pointing out are two particular circuits that we tend to focus on. Why? Because of the two circuits that are predominantly involved in movement. Now, the science is taking off in terms of other circuits that involve the movement. There are other circuitries, but I'm just highlighting the ones that we focus on the most. What you'll notice is there's two colors. One's in red and one's in blue. The one in red is really hitting the more familiar circuit that we talk about, and that is the circuitry within the basal ganglia that interacts with the surface of the brain at the level of the somatosensory cortex at the top of the brain. That automatic circuit is one that we think about as basically one you don't have to think about, right? So it's automatic, right? What's important about that concept is that that automatic circuit is created through what? Practice, essentially practice. So, you know, you don't come out of your mom's womb like going, hey, mom, how's it going, right? And you're certainly not walking across the room. So the point is that that circuit is created through practice. So has anyone read Tipping Point from Malcolm Gladwell? Okay, so a lot of heads nod, right. And so what does Malcolm Gladwell talk about? Exactly that. Repetition is key, right? That's how learning happens. So we're not talking about anything new. In fact, pretty much everything I said at the end of the night, you're going to say, you know, common sense. Well, it's been taking common sense to get back to these very simple ideas. But the idea, again, is fundamentally that automatic circuit is practice. You get that circuit through lots and lots of practice. And so the idea that I am actually hand-waving and gesturing like I'm doing, I'm actually not thinking about it because I tend to do it a lot. And that's practice as well. All right. The other key important point, getting back to dopamine is a modulator of the circuit. Think about it as fertilizer. So you definitely need it to be able to do what? To secure and encode. Aspects of encoding and securing the connections, what we call at the level of synapses, is important. But dopamine is a modulator of that. Fundamentally, you need to practice. And the key point is there is no way in hell you're ever going to get good in German or Spanish if you drop dopamine in the brain. Think about that for a second, right? Dopamine does not create magic. It doesn't create something from nothing. Dopamine enhances what? Practice. So it's needed for the acquisition of learned movement, all right? It's needed. The second point, though, and this is recently, probably in the last 4 years, where we recognize not only is it needed for acquisition, it's needed for maintenance. So as dopamine levels are dropping, what's going to happen is that circuit's going to have lots of problems, and it does, right? It has lots of aberrancy within that circuit, and I'll show you what that looks like in a second. But aberrancy is what manifests as fundamentally as bradykinesia. So when we're looking at bradykinesia behaviorally, which is what? Slowness not just in speed, but amplitude, right? It's because of aberrancy in that circuit, right, that's causing that behavior. So I'm going to get back to the idea again. Circuitry is what creates behavior, okay? Cell loss at the level of dopamine is a modulator of circuitry, and so behavior is a circuit-based problem. Now, the other system that I'm pointing to you out in blue is another very important aspect of movement, and we use both, the red and the blue, all the time. So what is the blue important for? Well, the blue is coming from the prefrontal cortex, and we call that the executor. You can think of it also as your desktop. It's actually updating and managing things on a day-to-day basis. What's kind of cool about that is it's allowing me to take what I would consider more automatic movement and do what? Refresh it, optimize it, make it flexible, make it relevant to the environment in which I'm in. Right? So I always like to remind people, we don't walk and we don't learn to walk in bubbles. All right? We learn to walk in context. It's always contextual. Right? And so the prefrontal cortex is always allowing us to adapt a particular skill to a new context and optimize. Perfect example is the Olympian, right? The Olympic diver. So he gets really good at making things automatic, but he's also using a lot of environmental cues to make that, very beautiful. You could argue that what makes us very human is really that prefrontal cortex that allows us to really refine movement in a very fluid fashion. So the point is we're using both all the time. We rely heavily on the automatic circuit, certainly, but the prefrontal cortex allows us to adapt and optimize and even do what? Dual task. So all athletes would be very familiar with the idea of dual tasking, or if anyone saw the NBA finals, right? Stephen Curry doing all he needs to do, right? I mean, that's dual tasking right there. So you're obviously concentrating on that move, but you're also able to adapt in the court. And so you're using both. So what do we know about Parkinson's disease in this context? And again, I'm leaving out a lot of other circuits. We're going to keep this story simple. As dopamine levels are dropping, I already told you there's problems in the automatic circuit. Now that circuit isn't completely gone, but it is not happy. What happens is patients become more reliant on what? The prefrontal circuit. So patients will tell you all the time that they're using their prefrontal circuit. They're not saying, I'm using my prefrontal circuit, but they're certainly saying, I'm having to do what? Think more about what I do. And that's really what they're doing. They're very prefrontal dependent. And so, that's fine for a while. And that's some type of compensatory strategy for a while. But the truth is, you think about how you'd get around your day if you had to do that all the time. It's very fatiguing, for one thing. It's not sustainable. And then you have the problem with, attentional demand and divided attention. You get told, someone's yelling at you from across the room, and suddenly your prefrontal cortex, which is already in high use, is suddenly being diverted, and what do? Fall. So these are very important concepts to think about as we think about what we're targeting. So one of the things I want to talk about too as we go forward, I don't actually study devices, I study concepts, right? So when we talk about biology, we're trying to drive hypotheses, biological terms, in terms of what is it that we think is going on. So I'm just laying out the field of what it is that we think we need to be addressing. So we're talking about an automatic circuit, and we're talking about a prefrontal cortex, and we're talking about how we can use lifestyle, in this case exercise, to try to impact these circuits in a way that may lead to repair. So then let's talk about the idea of plasticity. Plasticity came out way before the decade of the brain, all right? This is Ramon and Cajal in the early 1900s who had already been talking about that, and then it sort of went away for a while. But in the decade of the brain, in the 90s, what it brought back was some basic concepts, which is the idea that your brain is always changing. Now that's a good thing. Really? Do you want your brain to be like a 5-year-old? I don't think so, right? So you want changes happening. And the point is that when your brain is changing, it's always changing in the context of what? Your experience, of course. Exactly what I'm telling you. So it's changing constantly in respect to the experiences that you have. And so one way to conceptualize this, and I'll talk about the definition of plasticity in a second, I actually think about plasticity, experience, and lifestyle as what? A counterbalance, you see. A counterbalance to what? To everything else that wants to hurt you, right? Genetics, environmental toxins, whatever the hell. You try going through the list and curing everyone, all right? How many lifetimes would you need to be able to do that? That's not even feasible, all right? But what is feasible is creating a resilient brain. And what does resilient mean in the big scheme? The idea is it's making you what? More durable. And the best analogy that I can think about is leaving a harbor and crossing the Atlantic Ocean. So crossing the Atlantic Ocean, you're going to get lots of hits, whether they're storms or whatever, right? You want to make your ship as resilient to those storms as possible. It's not going to say that you're not going to get a storm. You might well hit many storms. But what you're trying to do is create a bigger ship, right? And so what that is modifying, isn't it? And the reason that's such an important concept is there's this tendency out there to think that once you have a disease state that somehow you fall off the cliff and nothing's relevant, and that is BS. All right? The brain doesn't know what it's been going through. All it knows is that through experiences it can do what? It can adapt. And it will continually do that. It doesn't know any better that you've decided because a person has Parkinson's disease, there's nothing to learn. BS, right? That's not true. So the point is that this is a spectrum. We need to think big. I mean, get out of the box and think big. The brain is plastic. You put experiences in front of it, what are you doing? You're creating a modifiable condition where you're what? Counterbalancing. And that is really how lifestyle fits into this picture, okay? All right, so let's move on and let's come up with some ideas of what neuroplasticity is. And fundamentally, it's the idea that the brain reorganizes itself by forming new connections, and it does that throughout life. And again, driven by what? Experiences and exposures, right? So fundamentally, where is that occurring? Well, it better darn well be happening at the level of synapse, otherwise you have a brain that we don't recognize. You're not human, okay? That would be the point. Because any human brain, it happens at the level of synapses. That's just the bottom line. And I was emphasizing this morning about, the whole idea of neurogenesis. I am sorry, it's cool to have neurogenesis only if it makes a synapse, all right? Having little brain cells floating around are not useful to you. I guarantee it. It does nothing. It does nothing unless there is a synapse. So again, neurogenesis, okay, show me a synapse and then it becomes relevant. Because really behavior is synaptic. And so you can also think about plasticity or neuroplasticity as a what? A type of homeostasis, isn't it? Again, the brain is compensating all the time. And that homeostasis that's happening is happening at a circuit level, what we call compensatory. And I gave you an example, patients with Parkinson's doing what? Compensating using the prefrontal cortex. Now, I would also argue that compensation isn't good or bad. it's neutral to the brain, isn't it? But it may not be neutral in terms of behavior. And so everything that we see in Parkinson's, some of it may be good and some of it may not be from a behavioral standpoint, but the brain is doing what it's doing. So what we try to do is think about guiding that and also constantly assessing, is this good or bad? Finally, two other main points, again, back to the idea that fundamentally plasticity is the essence of our brain function. Our brain wants to learn. You've got this amazing computer that's on a fire. It soaks up experience. That's what it lives for. It wants experience to do what? To constantly remold itself, to create some level of homeostasis. It drives on that, all right? Anything you don't do about that, I don't even know. That brain is so unhappy. So sitting in cubicles, yeah, it's a bad thing. Don't do it. And then finally, the idea that rehabilitation is doing nothing but harnessing this idea. There's nothing new. Everything at the end of the day, I'm going to say, duh. Really? Duh. Because it's simple things, fundamental things that the brain already does. So what are we really doing now is understanding the molecular biology of that. But there's nothing new here in terms of learning. We know this. This is plain neuroscience. We've known this since we've identified what is long-term plasticity that was done in the 1980s. We certainly know that encoding is really where behaviors are. And when we see disease states, what we're really talking about there is synaptic dysfunction, not cell dysfunction. All right, so these are examples of experience that we know, and we've certainly appreciated in our own children, as an example. Learning occurs. We certainly know that happens in athletes and Olympians. I mean, they're getting very good at a particular skill. And really, you know, in the last 10 years, 15 years, and our group was part of that whole big contribution, was the idea that we could use this not only in aging, and there's certainly a lot of aging field work, wonderful work that I'm not going to be able to talk about today, and in other fields as well, but that we can use these same concepts to drive plasticity, right, and to lead to some sort of repair in a meaningful way that leads to meaningful functional outcomes. And I think that's the other big ticket item certainly physical therapy brings to the field, and it makes us always hold true to function. You know, so it's not just about scales that we love, you know, and we have our little favorite scales, but at the end of the day, it really has to be about function. So there had been a lot of great work. So we were involved in some of the original work, and I'm going to show you some of the original animal work that was done. We did some of the original human work that was done. And since then, there's been a lot of excellent people in the field. It's a great field to be in because there's a lot of creativity in terms of things that people are interested in looking at. And what you see here is just a sampling of the different types of activities that have been done. And we've seen certainly a lot of benefits, particularly in gait imbalance. And of course, why do we care about gait imbalance? Because gait imbalance is probably the most important thing in terms of quality of life for Parkinson's and the things that we see most impacted. And so this is Bernie. I'm just going to show just a few pictures of patients before and after. I know you guys have seen a number of these. This happens to actually be my patient with Parkinson's disease. And actually what I just want to point out here is another thing that we see that is cognitive. It's a problem with cognitive processing. So because that prefrontal cortex is saturated, it has trouble updating and optimizing. And see, what you see here is when he's going over a different type of terrain, you can see that prefrontal cortex going, slowing right down. Like, please don't do this to me. Right? So he's on all cylinders trying to figure everything out, and I'm, right? So changing context is actually quite hard in Parkinson's disease as well, and that probably has to do with something with the cognitive processing being impacted. So we're saying, okay, so it's not just a problem of automaticity, it is a problem with the prefrontal cortex. And this is just an example of a big program. There's a lot of different programs. Again, I'm going to touch upon the idea of skill and goal practice in however way you want to do it. In this case, it was a big practice program. And what you see is his ability to perform a lot better. I always joke and say that if this was a stem cell, right? If this was a stem cell surgery, we'd be all over the news and we'd be funded up the wazoo. But, you know, our country is not interested in funding these sorts of things, but, you know, we go on regardless. Okay, and these are just some more new tapes by Claire. This is actually a patient with freezing. Let's see if I can get this going. Oh, come on. Hopefully, hold on 1 second. Let's try this again. Do I have it not working? Oh no, maybe not. Pardon me, one more time. Oh, I apologize for that. I'll try to see if I can get it back up by the end of the night. But anyway, it was just showing a patient with freezing and on the treadmill being able to recover from freezing. And he was involved in a lot of different skill gait practice to show that we can actually work with freezing patients as well. And let me see if I'm sorry, these were working earlier today, and I'm having trouble with this one too. Okay. Apologize for this one, but this was showing over the years, actually over a 2-year period with practice, again, involved in a number of different gait and balance exercises, being able to show jumping rope in a patient that wasn't able to jump rope. This patient had Parkinson's for 10 years. I'll go back to that in a bit to try to see if I can get it going again. So when we were thinking about getting back to studying exercise, so the biology of exercise, there's a lot of ways to study it, and we had to come up with a very particular hypothesis. And what we decided to do is take the learning approach. So we were arguing that exercise, if delivered in a certain way, is doing what you're learning. And you're learning particularly if you're doing it in a skill-based way where you're challenging them to do what? Do something different and do it harder. And the idea there was you could make skillful things harder by mixing it up or by increasing the intensity of it, make it over a time period, or making it harder by making it maybe the balance component of harder, making them reach longer, so there's different ways to make things harder. Now, the other important point of that is that as long as you're challenging a patient to problem solve, you can put them anywhere on the learning curve, you follow me? So if they're down here in terms of the disability, you match them down there on the learning curve, and you start with what they can do and build off that. And that's where you see very creative approaches to get people to do what? To walk. So fundamentally, our target is gait and balance, walking. Why? Because they can do that with practice and skill. And so we pretty much take everybody, wherever they're at. We take freezers, we take people in wheelchairs, we'll do everything to get them what? Up. So we get people mobile, and our full target is gait and balance. We're getting people walking. So that is always our end game, starting with wherever they're at. So our interest has been looking at evidence for synaptogenesis. Remember I told you about circuitry? Can we change circuitry? And that's one idea. The other idea, which is really mostly in the aging field, which is looking at all these peripheral effects of exercise, and we certainly know that exercise has a lot of peripheral effects. We know this, right? We know there's some effects on the cardiovascular system. We know there's effects on the immune system. So people have been looking at that and how those peripheral effects may be driving some of these central effects. What I'm going to walk you through, though, is some evidence that exercise is very interesting from the point of view that the brain is not a passive partner in this. So it's not simply this idea that you're getting all these peripheral benefits of some amazing nutrients that just covering the brain. It doesn't work quite like that, and that's what I'm going to try to show you in just a bit. I'm just going to walk you through the animal model. So we use MPTP. This was a human model before it was an animal model. So in the 1980s, there were some heroin addicts in Palo Alto that unfortunately decided to create their own heroin. Actually, it was the chemist's fault. These guys just got it. We should blame the chemist. In any event, they had tried to speed up the reaction of heroin and come up with this compound, MPTP. I've just shown you a little cartoon of how MPTP gets converted to MPP plus through an enzyme and gets taken up by these nigrostriatal neurons by the transporter of dopamine. It's sort of like a Trojan horse, gets in and kills the mitochondria and causes cell death. So we can actually emulate a type of model, if you will, of Parkinson's disease with dopamine depletion and cell death, and we did that in an animal model. So what we're showing here is basically some of the original work that we did, and what made this work so different was that we were interested in plasticity. So what we did was after we finished with cell death, and we knew when that happened, so cell death is done by day 5. There's no more cell death. Now we're looking strictly, because we introduced exercise at day 7. So I don't want a cell to come back. Please don't come back, cell. I don't want you to come back. I'm just interested in circuitry at this point, right? So at this point, this allows us to see if there's any circuit changes. Is that concept even remotely true? So we get animals running on a treadmill, and I would argue this is still quite skillful. I mean, how many mice do you see running on treadmills, really? But the point is that they also have to stay up on the front of the treadmill. So that is also skillful because they know what's going to happen back there. It's not going to be pleasant. So they better darn way stay up in the front. And we also throw in some healthy non-PD for controls, right? Because guess what happens in healthy brain when you exercise? It also gets better. So yes, it's a nice internal control. And what we see here are basically animals that are Parkinsonian. One has been running, basically for 8 weeks, 5 days a week with weekends off. And what you see are both Parkinsonian animal, and you can take a guess at which one had been running on a treadmill. Now, again, I always say, really, They've been practicing, they should get better, right? But it seems so obvious now, right? I mean, at the time I was like, oh my God, they're getting better. Yes, they are getting better. It's your brain in action. Guess what? If I run you a lot, you're gonna get good at running. And it doesn't really matter if you have Parkinson's or not. Isn't that interesting, right? Okay, so the other thing is we saw benefits on Rotarod, and we also saw, and I don't have this work here, Daniel Holschneider in our group also using the 6-hydroxy-dopamine lesion rat did similar work showing now improvement in other types of skills that were not related to a treadmill. So there was carryover to other types of skills as well. So it's not unique to, oh, I'm working on a treadmill, I'm just gonna get better on a treadmill. We saw generalizability to Rotarod, to balance beams, to some hand skill type of movements. So we had a generalized effect. So when you study Parkinson's disease in an animal model, you're gonna get asked about dopamine, right? Of course. The first question is, well, what's happening to dopamine? It's gotta be dopamine, right? Because we're, our whole world revolves around dopamine. Well, obviously, I'm trying to convince you to get out of that world. Think bigger. But our original work, we had to look at dopamine. I'm just showing you a dopamine cell here and all the fun things I get to look at when I'm thinking about dopamine, because it's important, obviously, for Parkinson's. I get it. But also what I'm throwing on there is some dopamine receptors, D1 and D2. And the reason we care, obviously, they're in the basal ganglia, but we've learned a lot about these dopamine receptors. And I'm going to say just a few words about the dopamine D2 receptor. Again, there's D1. For people who aren't Parkinson's specialists, there are two principal families of dopamine receptors, the D1 family and the D2 family. And the reason that's important is because they have distinct properties. D2 family, actually, we know a lot about in the last probably 5 years. That one in particular is very important for motor acquisition. My ability to learn a motor task, you need a D2 receptor. Definitely. And the other thing that's kind of interesting about that, you need the D2 receptor present to even maintain function. So if you block that D2 receptor, even after you've learned something, you're going to lose learning. You don't retain it without that D2 receptor working all the time. So D2 is pretty damn important for acquisition and maintenance of motor learning. And more recently, we're appreciating the role of D2 in aspects of cognitive processing, more importantly, inhibition. So we need the D2 receptor to do what? For mental flexibility. Isn't that interesting? Even though it's in the basal ganglia, it has these links with the prefrontal cortex that also is involved in cognitive processing, in this case, mental flexibility. So these D2 receptors are, and D1 have other properties I'm not going to talk about today, but the idea is that prefrontal cortex and the basal ganglia are very much linked. You should now begin to get that motor movement is not simply motor movement, it's cognitive, clearly. It has a very strong cognitive component, and there's some overlap going on there, isn't there, between the basal ganglia and the prefrontal cortex for these two systems. So let's look at dopamine and let's see what we've learned. You see two black columns right there. One of them has been exercised and one's not been exercised, and the take-home point is they're no different. There's no difference. So is exercise, the benefits of exercise happening because of dopamine levels? The whole response is no, it's not, okay? So if I sucked out all your dopamine levels and you've been exercising and the other person hasn't, there'd be no difference. So dopamine levels by themselves are not explaining this behavioral difference. However, we can do some really cool things with taking a brain slice, and that's what you're seeing up here, dopamine release photometry. You lay a brain slice down, and what you see are numbers where we've actually introduced a stimulation in these different parts of the brain to look at how much dopamine is being released when I stimulate. So we're stimulating dopamine terminals that are there. This is not where the dopamine cells are. The dopamine cells are down on the brainstem, but they have these long arms that stretch up. They release dopamine in these terminals at the level of circuits. So the circuits are up here. The dopamine terminals are there releasing dopamine. I'm measuring how much is being released by exciting them. And I can capture that by this neurophysiological technique, and I can see how much is being released volumetrically, how much volume of dopamine is released. And I can also ask the question, how fast does that clear from the synapse or from the extracellular space? And what we're showing down here, I don't know if you can see it, but the take-home message, if you compare white to light gray, the white is all exercise, light gray is non-exercise. It's basically comparing Parkinson's exercise and non-exercise. The take-home point is that there is more dopamine being released. So while the total level is the same, and that's irrespective of where dopamine is located in the synapse, extrasynaptic space, that doesn't change. What does change is the availability of dopamine. So with the same dopamine levels, that's what's happening. Your dopamine is becoming more available. So when you have those patients that are telling you, I kick in better after I what? Exercise, after I get on the bike. Now you know why. So dopamine release starts becoming more abundant, So whatever you have is being released, and it's being cleared more slowly. So it's out in the synapse for a longer period. And what we also see is that the transporter, which is what's being shown here, MPTB day 35, what we're comparing is no exercise animal, a slice of his brain to the exercise. You see with exercise, the staining goes down. That actually means less transporter. The reason we care is the dopamine transporter is what clears dopamine. Like a vacuum cleaner. If there's less transporter around, that means the dopamine is more available. So again, all these tricks that the brain is doing, taking the same level of dopamine and making it what? More efficient for you, right? And so while it's doing that work, it's also doing something on the dopamine receptor side. And this is particularly on the D2 side. I'm just going to point out, so these are two ways that we looked at the D2 receptor. I'm just pointing out it's mostly D2. You see this in this bar graph here, which is looking at the gene expression, and in that bar graph, which has all the colors, black, it's going from this low black bar to this high black bar. That's exercise right there. The D2 receptor expression is going up. We did not see it convincingly in the D1, but we did see with D2, gosh, golly, that is good for reasons I just explained. It better darn well go up. Yeah, we're going for motor acquisition and we're going for motor retention. I want that D2 receptor blazing, and it is. So we had to convince ourselves, that this was happening in the brain by using a ligand, falipride, that's now being used more commonly. It was originally used in schizophrenia, but used in Parkinson's disease. And we started off with the animal model. This is just a kind of fancy scan that basically takes a PET image. A PET image measures chemistry. So what you see in red is a radioactive label to the D2 receptor. It's superimposed on an MRI brain of a mouse. And that's what allows me to see the brain with the colorimetric. The color is the D2 receptor, and what you see is the animal brain from the MRI. The sailing is what you should normally see, lots of D2 in the striatum, as you would expect. But what you see with MPTB, this is a Parkinsonian animal, you see a loss of D2. So loss of D2 could partly explain why motor impairment happens. It's not just because you don't have enough dopamine that disturbs the circuit, but also the actual receptor, the docking, isn't there either. So you've got kind of a double whammy happening that's causing problems in that circuitry because you're losing the modulator of that circuit, you understand? What we see with exercise, though, is we see that it was coming back up. And so that was kind of cool. And this, I always joke that my animal would hit the top cover of a magazine before I would, and sure did. This was on the front cover of Movement Disorders. And so we followed this up with an exercise study in humans, which we were not as lucky in getting it funded for reasons I don't even want to get into, just call it ignorance. But the point is that we basically took falipride, and again, we asked patients to participate in a study that they were de novo, and we did not let them take even a cough drop. Okay, deal with it. Not a cough drop, not a Tylenol, not nothing, because there was no way in hell someone was going to start coming back to me telling me, that's the drug, forget it. So we made them just say, look, you're exercising 8 weeks, you can handle it, and now we're imaging your brain. Let me tell you, these patients were amazing. I want to give them all medals. It was 3 hours in the scanner. And that's not easy, with an amazing coordinator who did a lot of entertaining in ways I don't even know how to keep them in that scanner. But the point is that what we saw was an upregulation of D2 receptor as well. What you see in red is a subtraction scan, post minus pre. That red means upregulation of D2 receptor. This work has been replicated in sort of another group, and not the traditional group I would have thought about, addicts, whatever. Yeah, so they repeated this finding in addicts. Actually, it's methamphetamine abusers that they put in an exercise program. They saw the same thing, D2 receptor went up. Interesting in that case, because the idea of an addiction is that D2, remember its normal role may be inhibition. And so in addicts, the idea is they have loss of inhibition. They can't inhibit behaviors. So now they're linking increased D2 in the striatum to improvement in addiction, and they're beginning to see that finding now. So this has been this work of exercise in D2 has been seen now also in addiction group as well. And John Stossel in BC has a study going on right now to replicate this work. All right, so I'm not going to bore you too much other than to say we also looked at a lot of functional changes within that circuit. So this has nothing to do with dopamine per se. It has to do with what remaining circuits. So I told you when dopamine is depleted, that circuit is aberrant. We actually call it hyperexcitable. And what's the key player there in that circuit has a lot to do with glutamate. We can look at glutamate changes in a lot of different ways. Take-home point is that it's hyperexcitable, and it's hyperexcitable for a lot of different reasons. Some of the reasons I'm pointing out in a cartoon, it turns out some of the receptors in the circuit are hyperexcitable. hyperexcitable receptors, and also, so I'm showing you there just changes in the hyperexcitability that go down with the receptor types. And if you see on the top, there's like these little vesicles with red dots. That's the amount of glutamate receptor, that glutamate that's being stored. There's a lot more there that may also be driving some of this hyperexcitability. With exercise, we have evidence in multiple ways that we see less glutamate, less hyperexcitability within that circuit as well. So we're not just seeing changes in dopamine modulation, we're actually seeing functional changes within the very circuit itself. So what else have we learned? Well, I mentioned again, in that aberrancy, it's not just about what's happening functionally within the circuit, so how those circuits are communicating with each other, which I said was hyperexcitable, but also morphological changes. So when we talk about morphology, what we're talking about, physicality. What do those synapses physically look like? And what we know, as I show you here, is a example of a synapse, is they literally, with dopamine, they fall apart. They literally fall apart. In fact, they literally disappear. It's pretty ugly, frankly. So it boils down to the same concept again. We fixate on dopamine, but the truth is what's really happening is happening at the level of these circuits that lead to behavior. And so when we started looking at these exercises I've shown you, we've seen changes in dopamine neurotransmission, we're seeing changes in function, restoration of remaining synapses, which start off aberrant and become normalized with exercise. But we've done more than that. We're beginning to see changes in actual connections themselves. We've shown this in a number of different ways. The first picture I'm going to show you is kind of a fancy picture of a cell within the striatum, the basal ganglia itself. And what one of the grad students did was basically, he fills the cell, and then he draws it all out. So he has to do that a lot, believe me. It's one whole thesis project. So he survived the project, but it was a lot of work. And then what we was able to do was start comparing, and I'm just showing you an example. Here's what a saline cell looks like. Look at all the wonderful arborization. Now, the synapses are going to happen off of that. Think about little twigs that are going to happen off of this. But fundamentally, on the cell, the way it's receiving connections, synapses, remember this is a circuit, so it's receiving information from the cortex, surface of the brain, to close the loop. So it has all these amazing dendritic arborizations, right? Look what happens when you dopamine deplete. That is not one happy tree, right? You've slimmed it down in a big way. So you can see things retracting, right? Less context, less arborization. And what we see with exercise is more arborization, right? Okay, that's nice, right? Let's look at synapses though. So when we look at synapses, and this is just showing you how we can do it, we fill cells, that's what's in black, they're Golgi stained cells. We blow them up, and you see in B, that's one cell, and that's a dendritic arbor, that's just one arm coming out, a long branch, and we take a segment of that, and now you're seeing in C literally a segment, and we count. And so we do kind of a density. We do how many synapses, like thorns on a tree, per millimeter, okay? Think about that for a second. It's a hell of a lot of work. Anyway, it's been done using Golgi, and we've used different stains to separate D1 and D2 and look at those questions as well. Take-home point is really here on the bottom right corner. This is a dopamine-depleted animal with exercise, and if you compare it with on the left-hand lower corner, MPTB, that's just pure dopamine depletion. So what you see is evidence for what? Synaptogenesis, right? So that's also kind of exciting because now I'm showing you that exercise without changing dopamine levels has caused changes, right, that are morphological, physical changes, right, in the synapse, as well as functional changes. Dopamine replacement therapy doesn't do that. We know that for a fact, by the way. If you want to see the paper, I can show you the papers. Dopamine replacement does not cause this in any way, shape, or form. And are we surprised? No. How many people can learn math if I pour dopamine on your brain, right? Really? I don't think so, right? It's called practice. You have to put the time in. So it's a very important concept because what it does is puts dopamine on the back burner. It doesn't say that it's not important. Okay, clearly it's important, but it's not sufficient, right? So the brain responds to practice. You have to practice. You have to move. You have to do walking to get better at walking. I am sorry, but like I said, you have a human brain, all right? You don't have an alien brain. You have a human brain, and you have to face that fact. Practice is key, but we know that already. There's no surprise here. I think what people are shocked is that somehow that your brain hasn't fallen off a cliff because you hit 55, right? Or because I have, a headache, suddenly my brain doesn't want to work. It just isn't like that. The brain doesn't know that. It just knows what it knows, and it knows that if it gets experience, it's happy. The question that we ask is, what's the limitation of that? How hard can we drive that? you see, and that's still not known yet. So the next question we ask is about type. And we are interested in that because we did believe conceptually that skill practice or goal practice, making things harder and learning should, if I'm making sense of this brain, you know, then we should be tapping into cognitive processing. Duh, right? I mean, my cerebellum is going to problem solve to some degree, right? But not a whole hell of a lot, right? So we kind of know something about the brain, and we kind of know where problem solving, cognitive processing is happening. That's pretty much a prefrontal cortex. Okay, throw the dorsal attentional network, whatever. But it's going to be the prefrontal cortex, right? And so if you're learning something, you probably are using the prefrontal cortex. That's not surprising. So guess what? If I put an animal through a pretty tough course of skill, should you be acting the prefrontal cortex? I would hope in hell you are, right? Oh, yeah. Compared to what? Compared to a 24-hour fitness looking up at Fox News. I'm sorry, Fox News people. But yeah, right? Yeah. So if you are, you know, not brain responsive and you are on a treadmill, right? Then yeah, your brain is not going to look half as good as if you are skillful and practicing gait and balance in a meaningful way, right? So let's do it. Let's see what that's about. These are just examples of skill like I need to tell you guys, right? Like I need to tell you what skill and goal practice is. I hope not, right? You guys know this, right? Okay, these are examples, but I didn't need to tell you that. Okay, so let's put you on a motorized wheel. Okay, but these aren't just any simple wheel. On one side, what you see here is a smooth wheel, right? So at this point, all that animal needs to do is just hope in hell he doesn't fall off of it, all right? That's all he needs to do. He needs to focus and stay on that damn wheel, because it's going to be moving. And on the other side, what you see are spokes removed. Okay, that guy is going to be a little stressed, maybe, just a little stressed. And by the way, yes, we've looked at court levels, and yeah, court levels don't change, so it's not a cortisol-based problem, okay? Now, let's do some blood flow studies. And what's kind of cool about the work that Daniel Hochsteiner does is that blood flow studies are done with animals awake. They are not sedated at all. These animals go through 8 weeks of that training with the speeds climbing harder and harder. Isn't that beautiful? Isn't that what we're talking about? You make it harder. You get good at something, you just make it one notch harder. If you're not sweating, if you're not challenged, are you learning? What's the response? No, you are not learning. The way you learn is through challenge, making things harder. And so, with that animal, as I showed you, we make it harder yet, all right? So, after that practice, we put them on just in this case, they're just walking, and we put C14 iota antipyridine long-term to look at blood flow. And now the question is, where is that blood going? All right? How many people think that aerobic is going to be higher blood flow with aerobic in the prefrontal cortex or higher skill? How many people think higher aerobic? How many people think higher skill? Yeah? Yeah, okay. So, and some of you guys are going, what the heck, right? Okay. So remember, again, the idea is prefrontal cortex is executive system. It is our problem solving. If I have to problem solve and work through problems, to move through space, the prefrontal cortex, in theory, should be activated the more I use it. And so the take-home message is, yeah, it does, in a big way. So what you see is blood flow studies. This is probably the best data out there. In fact, it is the only data out there. Yeah, it's the only data out there that basically shows evidence for circuit-specific effects with exercise. Honestly, it is. And this is not just a small thing. This is a huge thing. Why? Because it's suggesting that the brain is what? An active participant. It is not passive. Are you kidding me? Please, stop. Yeah, don't say that to me. It is an active participant. It is not your heart. So for all those heart people that want to put people on stationary bikes, good luck, right? Fine. Because maybe your heart doesn't have to think about it, but your brain does, all right? Your brain is most definitely active in this process, and that's relevant. And we love that. Why do we love that? Because we can target, you see. Now we can play some interesting games here to try to do what? To target areas of interest to us, you see. And so if I'm telling you that the more skilled you are, we're driving more top-down cognitive circuitry than we would with aerobic. Now, I'm not fully dismissing aerobic, so forgive me for anyone out there in the field that loves aerobic, and I do like aerobic. It's just different, and that's the point. what we see with aerobic practice is down here on the right-hand side, non-skilled aerobic, we are seeing some hits in the PFC. We're also seeing some hits in the CPU, which is subcortical, basal ganglia. So, and we have some human data to back that out. So basically, we are seeing probably, honestly, what there probably is going to be, and again, I hate to get ahead of myself, but I'll get ahead of myself. The bottom line is probably with aerobic, we see more subcortical bottom-up effects. Isn't that interesting? And what we see with more skill practice is what top-down cognitive processing, the big pause button, right? We always talk about that. The pause button, that's important for what? Everything, basically, everything, right? Whether that is emotional regulation, right? Right? And also execution of movement through space, yes, right? Top-down processing is a biggie. It's a huge, huge thing. And probably hitting what? Attentional networks, so we're talking about things like any disorder that causes attentional problem, and just name a whole bunch of them because there's a whole bunch of them out there. So it's back to the idea that it's not that aerobic doesn't have effects, it does in a different way. And it may be more bottom-up basal ganglia versus top-down cognitive control, which is really heavy what's the top-down processes? Is attentional demands. Who won't want better attentional ability? Yeah, everybody here, right? Attentional ability should be much better, as well as working memory. So working memory is a very top-down process. Things like, however, mental flexibility. We all know some aspects of what mental flexibility is, our ability to handle changes in scenario, which is also pretty huge for us, right? Adaptability is part of that mental flexibility. That may be actually more basal ganglia, interesting, because of the D2, at least plays some role in that. So I'm not saying that the aerobic couldn't even have some benefits on aspects of cognition that may be specific to basal ganglia role, like mental flexibility. But the top-down effect, which we're all into these days, right? Mindfulness, isn't that what we're talking about? That's all top-down, right? Skill may be more relevant for a top-down processing effect. Okay, so what time is it, guys? What is it? 10 after 7. Okay, I'm going to wind up here with just a few other points. I just want to show one factor that we got interested in, because I'm going to go back to the idea that, so now I'm telling you that exercise isn't passive. It isn't just peripheral effects, and people are studying peripheral effects. Certainly growth factor, you guys have heard that, neurotrophic factor. All these very interesting effects that exercise may be doing in the periphery that may be helping the brain, right? Okay, now I have to come up with a mechanism. Think about that for a second. What the hell is going on in that neuron that would be driving this effect? One of the things we kept coming back to was what happens as I drive activity at a synapse. This poor neuron is doing what? It's needing more energy, more fuel. Metabolic demand goes up, you see? And so we were looking for a gene that might be upregulated by a stressor. A neuronal stressor that could also drive metabolism. One of them is HIF-alpha. HIF-alpha is hypoxia ischemic inducible factor. It actually gets upregulated with low oxygen levels, but it also gets regulated when the neuron is under a stressful condition, when it's not meeting what its metabolic demand. So let's go back and imagine ourselves as a little neuron in the brain, and we're working hard because we're part of that prefrontal cortex, and we're working really, really hard, and we're going, wait a minute, where's all the fuel? I've just used it all up. It starts upregulating this transcript to say, I'm in high metabolic demand and I'm not meeting it by what I have. HIF does that. And so people have written about it. It's called a hermetic response. People are studying that in the diet field. You know, this whole starvation diet. What do you think that's about? That's really what it's about. It's about a neuron being required to act in the context of insufficient fuel. And when the neuron goes through that type of effect, what happens? It changes its metabolic profile very, very fast. And what's kind of cool about that is products such as HIF, which is an immediate early transcription factor. That means it's turning on genes. That means it's activating and translating genes to new proteins that are relevant to help meet the need. Now, what's kind of cool about HIF is I'm showing right below that, which I don't know that you can see very well, but HIF is a transcription factor, as I said. It regulates other genes, creates messenger RNA for these other relevant genes. What's downstream of that are glycolytic enzymes, glucose-related enzymes, also factors that turn on angiogenesis, increase blood flow, and growth factors. So it's like hitting, you know, the big kahuna, right? You couldn't get a better transcription factor. It basically turns on everything. It's like full blast. I need more blood flow. Let's see. I need more fuel. Let's throw some growth factor in there, and HIF can upregulate that. So where's the evidence that this is happening? So we are beginning to look at this in a circuit-specific way. That means we look at areas of the brain where we see changes in blood flow and areas of the brain where we don't, and we look for HIF expression to see if it's upregulated. And what we do is we take part of the striatum, the basal ganglia, where we saw lots of blood flow with exercise, and we start doing a time course. What you see is elevation of expression of HIF as early as 1 day. There's no exercise. What happens 1 day? It's already upregulated. By day 5, it's already upregulated. So the animal's running for 8 weeks. We're already seeing it upregulated that fast, right? By day 5, and actually we see it come down by day 10. So it's turning on very early, with the idea being that it's now regulating other genes that are now turning on, like a domino effect that's happening, right? And so we're trying to see some of these other genes upregulated, and sure enough, we're seeing glucose transporter, we see upregulated by day 5, a little later. We see another part of the glucose metabolic pathway, lactate dehydrogenase, a little bit by day 1, but really by day 5, and even VEGF. There is the blood flow. VEGF regulates angiogenesis. We see it upregulated. So this is what we're working on now, which is trying to understand this idea of a type of metabolic stress. By the way, the cancer field, this is what they study. Cancer is a metabolic stress, and it actually causes some of these same things to change. This is why cancers are so rich in blood flow. It uses these same metabolic systems to basically drive these crazy changes with cancer. Turns out some of these things are actually relevant when it comes to synaptogenesis. Isn't that crazy? So the cancer field is really what we're looking at right now to help us understand these mechanisms. And the diet field, looking at starvation, and also what's the other big one? Ketogenic diet, right? You guys have heard about that. What the hell is that about? Ketogenic diet is using this hypothesis, the same idea. Ketogenic diet creates a metabolic stress in the cells. And in doing so, it starts changing the metabolic profile of a cell that's considered more healthy, even leading to synaptogenesis itself. Okay, I'm going to end on that note because I'm going to take some questions. I'm just going to, pardon me, I'm sorry, I have way too many slides. This is a group, as you know, it takes a village, and it takes a lot of people to do this work. I'm always so grateful to get a chance to speak. I always go back to the lab and say, hey guys, they loved it. And I get a lot of the graduate students are always so pleased when they hear how much they are able to contribute to patients with Parkinson's disease. We always like to thank people that we've been collaborating with. And as you can see, it takes a village. This is me learning to serve with my very patient collaborator, John Walsh. Mike Jackwood, my husband, is also up there at the top. He's also a pretty amazing hockey player, Canadian, you know, those Canucks. But anyway, thank you so much for your time. And I don't know if you want me to take a few questions now, Peter, or wait? Okay. All right, any questions, or you guys want to figure this one out a little longer? Figure it out a little longer? Okay.
I got some questions for you, but
Okay.
that's for later.
Okay. Here we go. I'm going to give you. Do I hand this off to him, or do you have your?
No, I'm all mic'd up.
Okay, perfect. Okay, so this is your switcher, but let's put this in first, maybe. Pardon me for a second.
Sure. So there, oh, yeah, I can pull this off.
Do you need that still, or?
No.
Okay. So let's just go like this.
This is the remote.
Yeah, we're going to put that in a second here. Let me just get this in. Nope, where was that one? Am I getting this? I know I have this in here. I'm going to ask one of you guys to see if you can slide this in here. It's a little tricky because it's tiny.
What's this?
Ah, here we go. I got it.
We're in
Yes,
there.
I did. You want it on that side or this side?
Doesn't matter, I don't think. Does it?
You can get it in there?
No.
I think it may fit in better here.
There.
Oh, you got it. Yay. Okay, I just want to make sure that, okay, it should be okay. Yeah? Okay, I'm going to get out of your way here.
And this. What do I do here? Projection here? Voila. Okay. Well, thank you, Giselle, for a very nice talk. My topic here is far more pedestrian. It's going to be simple molecules, and in fact, I have to apologize in advance to those of you who are not clinicians prescribing medicines for Parkinson's, because this will dwell very much on those topics. Unfortunately, those of us in the trenches taking care of patients can motivate those to go ahead and exercise and get to rock steady boxing or Tai Chi or dance or power or LSVT big, but we also have to push pills on them. And I thought what I would do in this talk is give an overview of the challenges we have in the clinic to deal with everyday problems of advanced Parkinson's disease, and to talk about what's new, what's coming down the pike because of clinical research, some of which we've been involved in here at Henry Ford for the last 20 years. And in fact, a lot of the sophisticated pharmacological data I'm going to present to you, especially those who don't deal with this, is actually pretty obvious, pretty straightforward, pretty logical, because the challenge here is huge to take a drug and tame it to behave properly. And so what I'm going to talk about is what Parkinson patients experience often within a few years of starting medication. A miraculous improvement, something called the honeymoon period, and then motor fluctuations, phenomenology of not doing well for a few minutes here, a few minutes there, an hour there, sometimes a whole day. And these fluctuations in control, which can be very frustrating and disabling, are because of a drug that is so useful that we can't do without it, but which is untamed like a wild horse sometimes. So we'll talk about the current limitations of therapy with levodopa, as well as some of the things that are coming from development of new therapies. It's worth pointing out that all of the problems of what we deal with today in medicine were foretold by James Parkinson, after whom this disorder was named 201 years ago. He has this very choice phrase in his monograph called the shaking palsy. Until we're better informed regarding the nature of the disease, the employment of internal medicines is scarcely warrantable. I wish we could write notes that elegant. But in any case, that is the fact. Until we have a good logic, unless we have some scientific basis for what we're doing, why bother? Well, fortunately, we do have with levodopa this simple amino acid that's present in some of the food we're eating back there, a medication that actually can remarkably reverse Parkinson's disease symptomatology if we give enough of it. If we medicate patients with several grams of it per dose, or with another medication that's mixed in even much less, we can get them to a good state. We've had this for almost a half century now. This medication is old hat. It's a generic. It's one of the most cost-effective drugs. And it's just a simple chemical that is actually the precursor to what is really missing in the brain, the D word that you heard from Dr. Petzinger's talk. Dopamine throws the switches, modulates the circuitry of the brain. It goes to different receptors, different parts of the brain. It has a key role in terms of reversing many of the Parkinson problems, although not everything, because there are other chemicals up there that are deficient. It has an enzyme up there in the brain which is in high abundance, ready to convert it, although that dogma has been reversed by recent reports that I'll talk about later with gene therapy that show that you can actually do better if you augment it. And so, although this drug, this simple chemical amino acid, is the most efficacious way of symptomatic, not curative, but symptomatic relief of Parkinson's, its clinical benefit can become increasingly irregular for some patients, but actually by 5 years, most patients. And by so doing, it does so in different ways. It delays its onset of effect in some instances, or wears off rapidly, or just gives no chaos to the onset of its effect. And this is very frustrating because it's so effective at sometimes that patients curse its name. And if that's the case, what can we do about it? Well, initial therapy, we really don't have to worry so much about the scheduling or the dosing. Even once per day in some instances will give effects. And I'll show you some of the data that shows why that might be the case. On the other hand, the time frame of just a few years into this disorder, which begins in the late 50s, so patients often have a 20-year career of having Parkinson's. The variability takes on different patterns. I'll show you those in a moment. And there are other strategies that we'll talk about. So these are the kinds of things that the patient's view of living with Parkinson's can be, let's say, at that 3 to 5 year time point. They have intrinsic features of this disorder that are frustrating. For example, as they take a walk, their legs start slowing down, take smaller steps, or they might have hesitation in getting going, or in repetitive movements like handwriting, their handwriting trails off. So these are motor fluctuations that are built into the disease, probably having a lot to do with planning and other aspects of the prefrontal cortex, or perhaps execution programs that we have in our brain that take care of semi-automatic activities. Whatever it is, someone's got to figure it out, maybe you. We have to deal with it, the fact that the patient can confuse that with what our medications do. And this variability in the second section here is the kinds of things that the drug and its therapy and its enactment of improvement can occur. There can be failure of medication effect altogether for reasons that we'll show that are quite logical. The pill never got to the brain. Or there is a delay, there's a long pathway, and in fact, after getting there, the turnover is so short. Beyond that, there are other phenomenology that we have to deal with in our Parkinson patients. The drug got there, they have a constant level of effect, and yet they still freeze in place, just like those mice on the treadmill that you saw. Something in the brain can shut down the ability of this chemical to throw the signaling effect in place, and it's like a transmission that suddenly lost its clutch. The patient is frozen, sometimes in very situation-specific circumstances, like coming to a doorway or being asked to name animals that have tails, as in one of our studies, or in other circumstances that are very frustrating but very predictable in a sense. And another kind of motor fluctuation, which I won't get into, are the involuntary movements in dystonia, which also add to the problems of living with chronic Parkinson's. Let me pass by that. It's just worth making a point that, like any drug, it's got to get from here to your brain, and it goes through a number of circuits. These are just the various steps shown on a diagram. The pill has to get out of your mouth into the stomach. It has to get out of the stomach into the small intestine. It has to get absorbed through a specific uptake mechanism. There are a lot of hurdles on this odyssey that can keep it from getting absorbed. And the fastest that a molecule of levodopa can get to the brain is about 15 to 20 minutes, given current technology. And yet, there is something coming that will make it faster. And the other thing that's been shown is by careful analysis of what goes on in the clinic setting, it's not only the delay in getting to the brain, which can be even longer than that 15 to 20 minutes. In one study, it was about 45 minutes in a group of typical Parkinson patients taking doses at 4 or 5 hour intervals. The dose can wear off fairly quickly. So that rise, oops, now I did it. Yeah, I can't see it. So the rise in drug effect is a fairly brief episode and sometimes pretty irregular at that. So patients have to get the drug in faster and have to keep it for longer if they're really going to do better, so it might seem. Well, one of the problems is this is an endoscopic view, someone who stuck a tube down for a view of the stomach 1.5 hours after a patient took a typical dose of levodopa, and there you see it in the folds of the stomach, unabsorbed, an hour and a half later. So one of the problems is our patients often don't take the medication the way they should, with a cup of water, and on the other hand, they might take it with a meal, also leading to the same problem. So this is just a physical issue. The second thing is shown by these diagrams, and again, I apologize to the non-clinicians, non-therapists, non-medication therapists in the audience, but the pill has to get from the bloodstream, once it gets absorbed into the brain. This is the blood-brain barrier. It gets transported by a mechanism that knows what it's doing. Once it gets there, you see circled the dopamine molecule. It only has about a 15-minute or so half-life before it gets chewed up by enzymes that are readily there to do that, and we really don't have really good drugs to slow down that process. So this is a challenge of supply-side economics, getting more regular delivery to the brain so that this chemical can be there and do the magic of signaling and running the circuits and making sure that Parkinsonian symptoms are kept at bay. The other curious thing about this drug as you analyze its effects is it has a very peculiar set of rules. On the axis going up, you can see finger tapping, and this is an analogy to slowness of movement. When someone can tap their finger faster, their Parkinson's symptoms are under better control. So the higher, the better. And then on the x-axis down here is the concentration in your bloodstream. So what this illustrates is not much happens until the blood level gets to approximately that amount in the bloodstream. So this drug has to operate with a certain concentration. Anything below that, nothing happens. So we can graph this out, and this is the kind of data that we've done in our clinical studies, trying to correlate a blood level and what happens. And there you see two kinds of information. One is the rise and fall of the drug in terms of the curve that sort of goes like that. And then on top of it is the finger tapping, the analog of relief of Parkinson's symptoms. So the drug level goes up and down, the drug is getting into the brain, and then the finger tapping lasts at an improved level for about 3 hours. So this is what a Parkinson patient reports to us. I took my pill, 15 minutes later it starts working, and it lasted for 3 hours, and then it wore off. So our typical response to that is, well, that 6-hour spacing between doses doesn't make too much sense. Let's switch you to 3-hour spacing. And sometimes that's the end of the problem, using that kind of logic. Unfortunately, there's a lot of chaos, and again, I apologize for the tutorial in blood levels and pharmacokinetics, but that's what it is. The long and short of it here is that this is a drug that has a lot of variability. Those up and down lines on the curve indicate that from dose to dose, different blood levels will occur. Sometimes you get there, sometimes you don't. This is a very imperfect drug. A really good drug would have vertical lines that are a minuscule version of what you see here. And on the right is an ingredient with levodopa that's supposed to give it a stable effect, and actually it arrives in a rather delayed fashion. So this is a terrible drug, horrible. Plus, it has to get to that level. You know, it doesn't work from the lowest levels. Everything below that line of the therapeutic effect is useless. So if you could design a worse drug, I don't know what it could be. On the other hand, it works pretty well when it's working. So this has been our challenge in research, to improve upon the blood levels and give a more constant effect. Now, usually this isn't a problem in the first few years. This is called the honeymoon period, or the long duration response. No matter what the blood level looks like, the brain has some way to hold on to the drug, to meter it out in some fashion that doesn't even have to do with dopamine, because the dopamine is long since gone by the time you hit the 2nd hour, the 3rd hour, and the 4th hour. And this little illustration of someone kayaking along on that curve of benefit looks just great, and the patient doesn't really have to keep their eye on the clock. Unfortunately, this is an illustration of what happens at 3 to 5 hours. The blood levels call the shots. In other words, if you can't maintain constancy at the therapeutic level, the relief of Parkinsonism goes away. So you'd think this would be a rather simple concept here. What to do? Hook up an IV line and run the drug in. And in fact, this has been done experimentally, and it works just fine. But unfortunately, this phenomenon of the long duration response really doesn't last for much longer. We don't know how to make it come back. I'm going to skip by some of these things. Here you see one of the drugs that we've had on the market. It was costs about $8 a day. It's called entacapone. It was a revolutionary idea because it extended the duration of action by a little bit. In the blue line, you see the blood levels on the left side of that graph, the blood levels of levodopa. And since you've now become experts in this, you can see that the blue line, the effect of entacapone, extends out the duration of action of effective levodopa above that threshold for a while. And the blue line on the right is an illustration of the clinical improvement. The patient on their rating of Parkinsonism gets better. The higher the curve, the more relief of Parkinson's they get. They get an incremental improvement. Is that worth $8 a day? I don't know. And unfortunately, that isn't the story. And one of the lessons I've learned is, don't take average data to heart. It doesn't tell the whole story, because average data looks pretty good with this, but this is, in the next slide, the kinds of things that go on when you really see real patients taking this medication. And it's a complicated curve. We call these spaghetti curves, but it's, in essence, 2 of the formulations that we use for trying to get more extended effect. Each of these graphs represents 2 doses of the drug at 4-hour intervals. The different colored lines are one of 17 different patients who have taken these marketed formulations, one of which has that drug you just saw, another which is a slow-release form of levodopa. And the message here is, you can see the number of instances where that dotted line, which is the threshold for effect, where the patient took the pill dutifully, and yet nothing happened because the blood levels were below the threshold. Nothing happens because the level didn't get high enough. So this is very disappointing when you've taken your pill and it's supposed to work and you paid good money for it, And yet this is the reality of current therapeutics, that we don't have really reliable medication effect, even though this drug works beautifully when it gets to the brain. So this is our challenge, and I'm going to pass by this kind of stuff. We have other drugs that don't work so well. Yeah, let's pass by this too. So suffice it to say, drugs don't work that well. But what we do have are some ideas that have come out of research and out of recognition of these limitations. This is a product that's actually marketed in the U.S. and elsewhere in the world, and in fact, before I was here at Ford, we were the first in the U.S. to work with it. This is Duodopa or Duopa, and the whole idea is take that medicine in pill form. The problem is the pill getting out of the stomach very often, so why not put it in liquid form and run it past the stomach, which has a escape valve that is often blocked, and instead run it directly into the upper small intestine where it gets absorbed in a continuous fashion using that pump system, and by so doing maintain a constant blood level. And a colleague of mine in Sweden who developed this shows that this concept works. Each of these colored lines is a different day of that patient. On the left side, you see the oral pills and all their failings with irregularity. You can see that each day is a little bit different, that there are high points and low points, and probably time below the threshold, even though they've regularly taken the drug every 4 hours. In contrast, on the right side, same patient getting a very constant level, and as a result, achieving much improved symptomatic control. The only problem is, to have this system working, you have to have a tube, like a feeding tube, placed in your stomach. You have a large pump. It's extremely expensive. Anyone want to guess the annual cost of this? $25,000? Going? $50,000? More. So, and it's just the same medicine that generically costs, maybe $2 a day. Same pill content, but now in a liquid form. So this is one of the reasons the cost of medication is so high. I'm going to skip by this. In fact, there's even data showing that it isn't that great. So we have another class of drugs that have been around for almost 30 years now. This is drugs that mimic the actions of dopamine, and there seem to be a lot of promise for these because they're very long-acting drugs. They are very selective. They don't have enzymes to chew them up. They seem like the perfect idea for therapy, and these are all the various versions of them with dopamine, the naturally occurring signaling molecule in the center there. And you can see that some of them have some homology to dopamine. They mimic the actions at the various receptors. They have more potency. They're more selective and are often used together with levodopa. By themselves, they don't work as well, but they do add to the consistency of effect, and they're one of the tools we have. Unfortunately, they have a lot of side effects. They can cause a lot of drop in blood pressure, or sedation, or vivid dreams, or hallucinations, and can turn people into gamblers, as well as having other behavioral disturbances. But at their best, they can help to improve control. And in fact, these drugs are fairly long-acting. These are just consistent blood levels that one of them has, and others are not as long-acting. So we have other tools, but they're not really the whole story. The best thing we can do is make levodopa a little bit longer-acting. I'll pass by this again. One of the options we have is one of these dopaminergic agonists is actually a useful drug to give continuously under the skin with a small pump. And here you see illustrated an insulin-sized pump with a subcutaneous administration route. We're currently using this in a clinical study, but around the world it's marketed as a therapy for that kind of patient who has the motor fluctuations. The molecule is sort of interesting. It's taking the narcotic morphine, reacting it with hydrochloric acid so it stops being a narcotic and is not addictive or having any of those properties, and it actually starts acting like dopamine, and so it behaves very well at improving clinical symptoms. And here you can just see some data that recently showed that patients were highly satisfied with it, and it improved their off time. So they found that their motor fluctuations that were otherwise occurring with oral levodopa was very much improved. And it's not that cumbersome to have this infusion system going. We hope to see this as a therapy on the market in the near future. What else have we got? Okay, so that's the current state of affairs. What I'm going to do from here on in, and we can go off in other directions of discussion, but I thought we could highlight some of the new therapies that have been under development for dealing with motor fluctuations. There's a whole host of ideas. I was talking to someone earlier today about the difference that Parkinson's poses to so many other neurodegenerative diseases. We have all kinds of great ideas of how to help patients, pills, infusions, and otherwise. And obviously, we can do some amazing things just by putting people in the right exercise and training programs. But the medications can do some remarkable things, and you can see what the challenges are. Well, here's some of the ideas that have come out of the pharmaceutical development programs trying to address some of the questions that you saw here. For example, levodopa is a prodrug, but you can actually make a prodrug of levodopa itself and lead to some improvements. Levodopa is absorbed orally and under the skin, but it can also be absorbed through the lungs. You'll see some data for that. Levodopa has the possibility of itself being given under the skin for absorption, much as apomorphine, and one of the studies we're working with has that as a theme. And then the next topics will be the idea of using alternatives. One of the strategies for treating Parkinson's that we have beyond medications is electrically stimulating parts of the brain that we know have improvement in store. And one of the pharmaceutical developments in recent years has been using a chemical receptor blocker that can at least address that same pathway where we now are putting in electrical stimulation as a therapy. There's also been a lot of development in neuroscience of gene therapy for various purposes, both for protecting against progression of the disease and for symptomatic treatment. And I'll show some data relating to the use of gene therapy as a way to change the chemistry of the brain in order to try and help with Parkinson control. And I'll finish up with a topic of freezing of gait, which has been very vexing. Patients who, even with constant levels of levodopa, can freeze up in very troublesome situations where they might fall. And currently a transcranial magnetic stimulation paradigm has been developed, which seems at least to have some promise for control of that. So let's look at a few of these. Well, one of the things that's going on with levodopa is the idea of maybe it could have some way of getting it better absorbed so it doesn't have that irregularity in the bloodstream. By linking it to a molecule that's similar to sugar, you can get a much wider swath of the upper GI tract where it can be absorbed. In fact, it can even be absorbed in the colon. So for a drug that has a very quick blip of absorption, as you saw in those curves, the absorption curve can extend over many hours by linking it to something similar to glucose. And in this study, this just is a cartoon illustrating the concept there. And actually, the product did very nicely in terms of giving a more constant level. Unfortunately, it was a little bit underfunded, the company that was developing it, that is, and went down the drain in the research bubble of 2009. But it's a good concept, and many drugs actually operate off this concept of having a carrier molecule that can help to get it into the brain. And there are others under development even in the Parkinson's space. The study that we're currently doing is using levodopa and the other ingredient, carbidopa, in a solubilized form. So instead of that tube that has to go into the stomach, we have a product under development and currently doing clinical trials, investigating whether infusing this drug rather than giving it in pill form can maintain constant levels. And the prototype for marketing might be something as simple as this patch pump, which is illustrated here. And this is actually an off-the-shelf insulin pump that could be used for administering this and other drugs as well, with a disposable reservoir and a little electronic pump system that can be moved around on the skin. Currently, we're at least testing out the concept of delivering this drug and finding some very good results for people who are having fluctuations. Nothing's perfect, though. It also irritates the skin in some instances. But the levels, you can see that these very constant blood levels are exactly what we're after, and this is something that is achievable by changing the delivery system with the same old 50-year-old drug. Another product that we've been working with is a concept of using another delivery system, the lungs. The lungs are very good at taking up small molecules, and this simple amino acid, levodopa, is something that can be very efficiently absorbed with breath-actuated breathing device, much as people use for asthma and other drugs. In fact, even insulin has been administered this way. The photomicrograph that you see there, these little microparticles look like popcorn, but they're mostly levodopa, and as you breathe them into your lung, there's the opportunity for fairly rapid and efficient uptake. In fact, Where'd the graph go? It got eaten up. Suffice it to say, the rise in blood levels is in about 5 to 10 minutes to therapeutic ranges. So it's a very remarkable treatment option as sort of a rescue for patients who are off, who if they take a pill will have a minimum of 15 to 20 minutes to wait, and even there might not get to that threshold. In contrast, because of the direct intake of something in the lungs into the bloodstream, you can get a very reliable hit to the brain within 5 to 10 minutes in most instances. So this can fill in for the potholes of irregular levodopa intake. Anyhow, the clinical effects of that we show here. This is from work we were involved in our clinic, and you can see how rapidly Parkinson's symptoms improve compared to the top line, which is placebo treatment. So within 5 to 10 minutes, there was some effect, and it increased at 20 minutes and at 30. So patients clearly were showing benefit in a reliable fashion. Another point to make in that upper line there, that's the placebo effect. And just about every study we've ever done in Parkinson's has a placebo effect that is, on average, somewhere in the range of 15 to 20% improvement on any scale we use, whether patient satisfaction, tapping rate, you name it, walking time. People have very good minds. Maybe their prefrontal cortices or other parts of their striatum are very responsive to the power of suggestion, the hope that they will get better, the extra gumption they have in participating in the study. Whatever it is, we always have to deal with this effect that maybe human nature is full of placebo effect as well. And in fact, anytime we prescribe a drug, we're also capitalizing a little bit on placebo effect, maybe to the tune of 15 to 20%. In any case, there it is, not as profound, though, as the effect of levodopa. And finally, as I mentioned, Parkinson's is more than a story of dopamine deficiency. It has a lot to do with it, and certainly you can get some great improvements early on in the disease. But after a while, the circuitry catches up with you, or perhaps other profound chemical changes happen. This diagram just illustrates a simplified view of the complexity of these reverberating circuits that are important for understanding how the brain works in the motor system. Forget the thinking part of the brain or the other complexities of Parkinson's, which have nothing to do with slowed movement, the non-motor features. Where we operate with levodopa and the drugs that mimic dopamine are those blue lines on the outside, and replacing the missing dopamine does a great deal of good for the patient. But we also have another part of the circuitry which is called the downstream, after the dopamine. And this circuitry is important because in recent years, well, the last 25 years, we've had the ability to interact with that electrically. So that's where deep brain stimulation, putting in an electrode and shutting down various elements of that, and actually improving a lot of symptoms like tremors or involuntary movements, or even gait problems that can't be helped any other way. This kind of therapy is invasive, expensive, and although very effective, it'd be nice to be replaced. And so one of the concepts that came out from basic neuroscience and translated to clinical trials has been using a targeted chemical to try and shut down that pathway without having to go through the efforts of putting in electrical stimulation. So there's a particular chemical in the brain that's called adenosine, one of many signaling chemicals, and there's a particular receptor of adenosine that's called the A2 receptor, which is found exclusively in a part of the brain that has to do with that downstream pathway. Found nowhere else. It's actually very similar to caffeine in terms of how it acts, but much more potent than a cup of coffee. And indeed, caffeine doesn't do much for Parkinson patients. But this compound, which is called istradefylline, does get to those receptors very selectively. Other compounds that have that same property are there, and yet don't seem to work as well, it seems. Istradefylline has been studied for about 12 years, and one of the frustrations of Parkinson's disease studies is some trials seem to be positive, some seem to be negative. The drug is marketed in Japan quite safely and effectively. It works well there. It just hasn't worked in North America all that well, except in our clinic, where we got some good results. And again, you can see how effective placebo can be. But what this data here, the dark green bar, is our study with this in patients who have up to 6 hours of off time, being under-medicated despite regularly taking levodopa during the day. And what you can see is that they improved by about 11% as compared to the placebo improvement of about 4% of this off time during the day. A meaningful effect. In fact, even an hour of improved off time for a patient is something that they can treasure in terms of improving consistency. But it's doing this in levodopa-treated patients by going downstream. So the concept that you can actually pharmacologically do what DBS does, I think, is supported by this. And it might be that using other downstream targeted drugs might also help to improve off time and other problems of Parkinson's, and maybe make electrical stimulation of the brain obsolete in the future if we can figure out better ways to make this happen. But it's an irregular effect. It doesn't benefit all patients, and I'm not even sure if this drug is going to make it, because some of these studies had far less robust effects than we see here. So now let's finish off with something that goes into the future, you might think, but gene therapy is really here and has been going on for more than 10 years as a therapeutic advantage for treating different brain diseases, especially brain tumors. In the field of Parkinson's, there have been several studies, one of which was actually looking to see if a gene therapy approach could arrest the disease. But I'll give you a little background on it. Gene therapy is just a gimmick, if you will, using the known property of viruses to infect cells for therapeutic purposes. And the viruses that are used for this are stripped of their pathogenic capabilities. And the adenovirus that's used in some of the studies I'll show you here is actually an upper respiratory infection virus, some of which we may have been exposed to. But this is a version of it that won't do any damage to cells or make you start coughing in your brain. Instead, it can be used as a carrier for engineered genes that are inserted in the genome of the virus itself. So the virus knows how to replicate, knows how to invade a cell, has structural integrity, but it's also carrying with it this Trojan horse, if you will, of something that is therapeutically intended, and it gets in a very controlled manner, replicates to a certain extent, and then gives up the ghost after a while and is cleared, and no evidence remains after a few days. So it's a neat concept. It can be given intravenously, and it might spread through the entire body, or you can put a probe directly into the brain and then release fluid with a known amount of this vector, as it's known, and the transmission of the neurochemical effect can be very much controlled. So that's gene therapy. So this has been employed for two purposes in terms of motor fluctuations. One of them is to increase the synthesis of dopamine. So you can take the pills and get more drug in your brain, but wouldn't it be nice to have a little chemical factory going up there? And that's been the concept of two directions of therapy. One of them is to look at this process of generating dopamine. And well, it's been thought that an enzyme that is called the decarboxylase enzyme is abundant and couldn't need any more. That's the dogma I learned in medical school. In point of fact, it's been subsequently found to be somewhat deficient as a disease progresses for various reasons. And if you could only put more of that enzyme in there, perhaps the same dose of levodopa would work better, give you more extensive on-time. This was worked out in animal models using what you saw earlier, this toxin MPTP and 6-hydroxydopamine. And sure enough, it seems tenable that this could work. It was tested out as a gene therapy experiment in animals as well. And we do need those critters to be able to show the proof of concept as well as the potential safety, picking the right dose. So animal research is absolutely necessary to go these directions. So such an approach was taken just to increase that one enzyme and see if things could happen well. And a study that's ongoing is then replacing this aromatic decarboxylase activity, just increasing it threefold over its endogenous levels, and by so doing, increasing dopamine synthesis. And the work that's going on with it right now is just reporting that it's been safe and efficacious. That's all that's come out from clinical studies. But we'll await to hear its final results. But an even more clever approach is going on in England and France with a product that's called Prosamin, and we might become one of the sites for investigating this. So not only is that enzyme being inserted with the gene with the viral vector as one of the genes, but another gene construct is the enzyme, which is the rate-limiting enzyme to create dopamine from two amino acids that are in our diet, tyrosine and phenylalanine. And there's a cofactor which is needed to make that enzyme work. So they stuck a little bit of that gene in there too. They got plenty of room in a virus called the lentivirus, which in its other life is a source of huge health problems in parts of the Southwest where it's a lethal virus killing people exposed to rats. Even rabies virus is being used as a viral vector for tentatively in some human studies. In any case, these three gene constructs going into the brain, increasing the generation of dopamine, has also shown some potential for improving patients. So this might become an effective therapy in the future. The results so far look promising. On the other hand, you could imagine this being increased by just better drug delivery. Who needs to go into the brain if you can get a more constant blood level? So in a sense, you can see the pharmaceutical industry battling one another, trying to come up with a simpler concept, maybe maybe more invasive, maybe less invasive. The goal is pretty obvious: more constant effects, less motor fluctuations, more constancy of effect. But somehow this very sophisticated molecular biology approach is still ongoing, and there's even more exotic stuff going on. People are still doing fetal tissue transplantation. There's a lot of interest, although no solid research, in putting stem cells in the brain to try and do this. But all of these things are all aiming towards a simple problem: why can't the patient stop having fluctuations the way they do in the first 1 or 2 or 3 years? So really the biggest challenge is to get some basic neuroscience to figure out what is it about that honeymoon period, what is it that makes the first 2 or 3 years with that constant duration effect, why can't it continue? And it does for some patients. Not everybody fluctuates. So we got to shake it out of some of those patients' brains that are having the good responses and find out what's making them tick so well to really make progress and forget all these pharmaceutical developments. Finally, we were involved in a form of gene therapy here, which was looking at another novel concept, and this is changing the neurochemistry of the brain. And in this concept, we used an adenovirus, same concept, but instead the whole idea here was to change the circuitry of the brain. So here's another diagram, and you can see the dark arrows on the right, the Parkinson brain circuitry is altered, and the thickness of those arrows indicates increased output of the neurochemicals coming out of that portion of the circuitry because they're missing dopamine. When you lose the dopamine effect, the downstream effect of that is to increase the glutamate effects, and the net effect of that is to screw up the circuitry, and Parkinsonian symptoms are part of that. We know that because if you go in and damage the neuron structures, the subthalamic nucleus, the globus pallidus, use electrical stimulation to shut them down, or if you just go in and burn them out, the patient gets better. But that's pretty invasive. Can we do that some other way? Well, our gene therapy study targeted this part of the circuitry, and the reasoning there is that if you go to the subthalamic nucleus shown by that purplish arrow and shut down the activity of that, which is putting out a chemical called glutamate, you might achieve the same thing and not have to have a patient hooked up with wires and so on. So the whole idea of this was to take the chemistry of that, which is glutamate output, and turn it into GABAergic output, changing one neurotransmitter into another, one that goes from having an inhibitory effect to a stimulatory effect. And that regional neurochemistry can be done with a gene therapy approach by just inserting a gene that makes that enzyme abundant there and take out the other enzyme. And so this study was carried out, and the clinical results are a little complicated, but the net result is patients showed improvements within 3 months and more robust at 6 and 12 months compared to the placebo effect, which was there. This involves surgically putting the gene therapy into both sides of their brain in the hopes, one side of their brain in the hopes of improving their symptoms, and then originally and then on both sides in our study. And indeed, there was improvement in a number of things. These indicators showed that patients had more consistent effect during the day, that they had less fluctuations. In essence, we were simulating what they got from deep brain stimulation. And indeed, when we did sort of a meta-analysis look at what kinds of improvements these patients had compared to deep brain stimulation, well, the degree of improvement, percent improvement in Parkinson scores were very similar to the average kinds of scores that they got from deep brain stimulation. The difference here being that you could do this as a one-time operation, no apparatus needed, much less potential expense, maybe much less risk. Unfortunately, this company is no longer getting financed for this kind of thing is another challenge. So I'll finish off with another form of therapies that is widespread throughout the neuroscience and psychiatric world. And this is using stimulation of the brain. It is not ECT and is not putting in wires, but rather just using very strong magnetic pulses with the electric fields that they generate in order to improve regional brain function. And in the treatment of depression, this is very much replacing ECT, electroconvulsive treatment, because without causing a seizure, one can nonetheless achieve many of the improvements for control of depression and other psychotic symptoms very effectively. But because of the anatomy and knowledge of circuitry like the prefrontal cortex and other important way stations in the symptomatology of Parkinson's, we also have the ability to change temporarily and sometimes for hours and days afterwards functions. And this is just very preliminary and new data in terms of using this for another kind of motor fluctuation, namely freezing of gait. A group in Tel Aviv has been recently reporting that by stimulating the brain for just a few seconds, one can get hours of relief in terms of the problem of freezing of gait, of coming to doorways and feet locking up, or every time you start to walk, being frozen in place. And so this kind of modality, which could conceivably be a home therapy, which could certainly be much safer than putting wires into the brain and perhaps might replace some of our medications, is also quite promising, because not only were there improvements in freezing, but even some cognitive abilities seem to be improved. So maybe we can get two for the price of one for the Parkinson patient. So I'll lead to the summary here, and we can open up to discussion with both of us. First of all, motor fluctuations experienced by patients can arise possibly from multiple causes and in multiple types, and in fact the same patient can be vexed by many of them. We can deliver levodopa better with current technologies, and certainly the future offers even better ways, and this will solve a lot of the problems that we deal with every day in the clinic. It's hard to imagine life without levodopa in treating Parkinson's, but I could certainly imagine life being a lot easier if we had more constant therapies that we could offer patients routinely. There are other adjunctive therapies that might be effective, so the idea of going downstream, of using devices like electrical stimulation or magnetic stimulation of the brain as a therapy might revolutionize treatment. Unfortunately, we still have a disease that progresses on its own, and we still have to find the cure for it, but at least symptomatic therapies are doing a pretty good job, and for our patients who are 10 and 15 and 20 years out, at least their brain has found some way to keep this condition at bay. So I think I'll finish on that note, but be glad to open this up to questions and see if there's any further discussion for Dr. Petzinger, or any questions any of you have on Parkinson's, and we'll make this as long as you want. We also have a few uncorked bottles of wine and some other beverages back there that need to be finished, so please avail yourself. And you can get up and stroll around here and finish off our tasty desserts as well. So questions, shout them out. Yes? Oh, let me go out there with the microphone and I'll.
Oh, I could just stand up and shout.
No. We've got a mic. Okay, yeah.
Thank you. So when it comes to the exercise in the Parkinson's, how frequently are you recommending, and what happens after, say, 5 years, 4 years?
Right. So how frequent do we advise it?
How frequently do you tell them to do, like 5 days a week?
Yeah, so these are all excellent questions. Most of the studies that have been done in exercise in Parkinson's have been traditionally about 3 times a week. Most studies have ranged from anywhere from 8 to 12 weeks as an intervention, as a single intervention. But the idea of everything we know about learning is, use it or lose it, use it or lose it. The idea is, yeah, you need maintenance. But you would need maintenance for anything, whether it's learning Spanish or anything else. So typically what I'll do is I'll get people into physical therapy about 3 times a year, 3 to 4 times a year, and I start right away. So right now I can honestly say, and Peter's heard me say this before, 50% of treatment of any neurological disease should be lifestyle. The data is that strong. If you're not advising physical therapy or exercise in your practice, you're doing something wrong. And we're very proactive about it. You don't wait for people to fall. So it's an element of your practice right off the bat. You talk about drug treatment, and it's a fair and balanced talk. 50% should be about drug, the other 50% about lifestyle options. You talk about how often. We basically say about 3 times a week, about an hour 3 times a week. I get them first into physical therapy. And the reason that's important, particularly with Parkinson's disease, is that they need a tool set to work on. And they can always get better, right? So really the role of the physical therapist is to help them target some very specific problems that they may be having, and then they can do all sorts of exercise things after that, right? And they have a tool set to use with anything that they're doing. So they're always in exercise. They're always doing something. And every time they come back, I tell them before they leave, I'm going to ask you. You better be doing it, because I'm going to ask you. So it's very unusual for me not to have patients that are exercising, as you can imagine. All my patients are exercising. Actually, for my patients, it's more like, what are you not doing? In fact, it's actually hard for me to do exercise studies because I get everyone's exercising. And variability is good. So the data is pretty strong. And again, the physical therapists in the field will certainly know this already. There's a lot of things that are known about what's called self-efficacy and self-belief. And that is being able to do something you love doing counts. So you absolutely want them to be engaged in something that they love to do, and that can be anything. So the only thing you need to be doing is making sure that you're telling them, look, besides the physical therapist, and I think physical therapy is important, I put physical therapy ahead of trainers. If they have some neuro-PT background, all the better. And I think you guys have obviously some experience, some people here that have that type of background, because they can really make a big difference in terms of what they're educating their patients and doing. So yes, they do that. I get them involved in that immediately, right off the bat, and then in an exercise program. I should also say, and fortunately where we are, we know a lot about the resources that we have in the community, so we're digging up resources all the time, whether they're dance classes, whether they're gait and balance classes, Tai Chi, yoga. Knock yourself out, right? Go surfing if you want to. I don't really care. Whatever you love to do, even better, because you have to have that. I mean, that's really important, again, the idea of self-efficacy and self-belief. You also like some variability, so that was getting back to the idea. Variability is not just in terms of the types of exercises that you're practicing, again, skill, but variability in the places that you're practicing. So, we didn't talk a lot about that, but the idea is there's a lot to be said for the environment itself. There's been some interest in studying that more, comparing nature, right? Doing things in nature versus doing things in a single gym. And certainly there's some hints there that having more environmental enrichment, that's called a cognitive load in and of itself, right? Because I told you when we move, it's not in a bubble, we're entraining that information as well. You can mix things up by mixing up your environment. So I'll tell people, look, if you're not in an exercise class, if you're not doing something with a physical therapist, then go out there and do some walking, and mix up that. Do it on a trail, right? Do it on a beach, do it, in a gym, do it in a mall. Doesn't that sound like Dr. Seuss, right? Green eggs and ham. I always tell him, he completely got that, right? On a plane, on a train. It's like that. You want to mix up the environment in which you're doing it. And so the bottom line is that you can start seeing right away how many things your patient can really do, right? I mean, you don't have to be, you can give them the big picture. Yeah, you want them gait and balance as much as you can. Absolutely. But after that, sky's the limit. Go out there and knock yourself out, right? And do stuff that you want to do and you're committed to. Then we get them back into physical therapy. Final statement, I often turn to the whole family and say, are you kidding me? You think this is PD specific? I don't think so. You guys all should be out there. Like all of you should be out there doing it together, because this is not unique to PD, for God's sakes. This is a brain that belongs to all of us. We all need to take care of it. So this is truly not a PD specific thing, which means getting the caregiver involved, getting everybody in exercise, because it's really a community resource, isn't it?
But do you lose the benefit after 3-4 years, like
drugs?
Would
you forget Spanish if you stopped practicing Spanish, right?
But even after doing it,
so what's the long-term retention? So we do have some data on long-term retention. That it's not our data, it's other people's data that's shown like up to 3 months, there's still some benefit, right? And there's also some animal data suggests changes. As an example, Neurotrophic Factor, which I said is one compound that people are looking at in the periphery, work by, in our Irvine, forgetting his name, Carl Kottman, has shown that that neurotrophic factor comes back faster on return of exercise practice or whatever. So it goes away, starts diminishing over time, and then comes back faster when you re-engage as one mechanism. So again, the idea is that, yes, you do lose some benefit over time, but when you restart, you can kind of kickstart that a little faster in terms of some biologics that are important, biologic mechanisms may be important. Nothing surprising from everything we know and learning. So pretty much I try to get people, I really get on them, But, honestly, if we made the effort that we talk about pills, think about that for a second. If you made your first question, instead of what are you taking, if you made your first question, what are you doing, right? How would that change people's behavior in terms of what their expectation is? And the other point is, if we as healthcare providers aren't doing it, why should we expect our patients to be doing it? What led to the biggest change in cigarette smoking in this country was when doctors stopped smoking, right? So those behavioral changes have to start with ourselves. We have to be the example. So it's hard to be talking to patients about exercising if we ourselves are not doing that, right? So that's, this is a challenge for us all, right? And the good news of this is that our brain just sucks this up. Our brain loves this stuff. We're talking about something that is normal for the brain. Everything we've been doing to date is not normal. Being in cubicles is not normal, right? That's the point. The guy out in the Serengeti with the little grass skirt, with his little spear, with the little, eating dirt clods periodically, that's normal, okay? He's got it right. You want to see a poster board? Put up the little guy in the Serengeti with the little grass skirt and his little spear and say, that's who I want to be, that little guy with the grass skirt. Because that is what we should be doing all along. All the behavior that we do right now has to be unlearned. Getting away from the TV when a patient starts telling me they can't walk, get off that stupid TV, turn the TV off, stop watching commercials, get off your phone and walk, right? Really, you got to have that mentality. You don't accept anything else. And if we created the same passion that we gave to pills in this country, are you kidding me? Yeah, it's a nationwide problem. In UK, if you want to get online and see some stuff that they're doing at the UK, they put us to shame in what they're doing in England in terms of physical life, blowing us out of the water. I mean, many European countries are blowing us out of the water. We're like the last learner in the group in terms of what we should really be doing. Even with kids, cutting out all that gym and physical activity, that's bad, right? I don't know what we were thinking, but we definitely are behind the times. And so I'm a big promoter in making us all active as a community. It's not a PD-specific problem. It's an American problem. Yeah. Yeah. Yes, there is. So epidemiological data, and actually there's some epidemiological data on caffeine as well. So what do we know about the relative risk of Parkinson's disease and Alzheimer's disease with exercise? Lower. It's lower. And that's true with caffeine as well. That may be A2A. I'm curious about that. But yes. So there's data, epidemiological data, that lifestyle does have some mediating effect on Parkinson's. Hell, for all we know, Parkinson's starts as a child, when you're born with Parkinson's. That's been thrown out there too. So it gets back to the whole idea. Look, if it's not one thing or it's another, are you kidding me? If it's not Parkinson's disease, it's a cancer, are you kidding me? Right? What, we're going to make it through the whole list? Come on. Okay, you can do that, or you can just say, hell, we have some things here that are quite powerful, and it's called lifestyle. And we're not even beginning to touch that in terms of its power for what? Resilience. And so there's been some amazing studies more recently in the last year. Some of them have even looked at ApoE4. These are risk factors now. Risk factors for Alzheimer's. And what does that data show? It shows it doesn't really matter what your genetic risk is. If you follow through with lifestyle, it kind of blows it out of the water. In fact, don't even bother looking at 23andMe, because if you're kicking ass in your gym, it's not really going to matter. I don't care what your genetic risk factor is. As long as you're putting the effort in on the lifestyle side, forget 23andMe. Don't look at genetic risk factors. It's not going to matter. And that's what the genetic people don't want you to know. Mm-hmm. But that is the truth. Lifestyle kicks ass. It does. Peter.
Just beyond that question, is there a dose-related effect? In other words, the skeptic says how much lifestyle change or how much exercise, how many times per week, and a dose-related effect would be useful to gauge that. In other words, does the elderly person really have to go to the gym every day, and is it 3 hours, or does this epidemiological evidence give us at least a threshold for seeing the benefit?
Yeah, and those are excellent questions, which is what the dose level is of that. And there is some work that's being done on that. Again, I would say most of the data to date has been following the American Heart Association recommendations in terms of the amount of physical therapy or the amount of exercise to be using. We've pretty much been using that, and then people have looked at kind of cutting that back to twice a day and then how intense it is, using different biometrics. So one of our challenges is trying to track that in terms of what do you call intense? Is that an hour thing? Is that the amount of hours? Is that how hard you're working out with your heart rate? There's still a lot of questions regarding what you call intense, because if I'm interested, for example, in more about skill training, I wouldn't necessarily be looking at heart rate to try to measure that as a metric. So I think in the field right now, we're still trying to figure out what those metrics are in terms of, is it a duration effect? Is it how hard your heart rate should be? Some people in the aerobic field would argue, you don't have to be working out for long periods of time. It could even be bursts of getting that heart rate way up there in the aerobic field for some of the data that they're looking at. So excellent questions. We don't really know that yet. These are studies that are ongoing. So there's data, remember, it's not an absolute, these are relative risks. So the idea is that caffeine, there's data with caffeine, and there's data with smoking. And obviously, epidemiological studies are not cause and effect, they're associations. So you have epidemiological data, and the next question is why, and what is that relationship? So the idea is, yes, there's been some data with caffeine. So who knows? Caffeine acts like an adenosine antagonist, what Peter was mentioning, and so does that have some long-term effect in terms of modulating A2A? Again, if you're a believer that truly whose brain is normal, really? I don't think so, right? There's no such thing as a normal brain. What does normal mean anyway? And do you even want a normal brain? I don't think so, right? You've got a brain change. Deal with it, right? You're going to have a brain change. It's going to happen. Aging is going to do it. We know that. And anything else is going to do it. Plus, if you live in a pesticide-ridden valley, which I grew up in, I should be in a full panic mode right now. I was like dumped on with pesticides. I lived in Sacramento Valley, one of the largest usage of pesticides in the world per acre, right? The idea is you can't avoid these sorts of injuries, right? But the idea is not to be alarmed. You have some other aspects of your body that can respond and become resilient. And so again, the bigger picture of lifestyle, I don't necessarily think of it as a cure per se. I think of it as a modifier and a counterbalance. And that's back to the idea of resilience and reserve. There are now studies going on, even like, for example, in the Alzheimer's field, where they're trying to construct ideas of what that really means in terms of resilience and reserve. What are those factors that we think are important that are related to what do you call dictators of resilience and reserve that are giving us some idea of counterbalance, that they predict, oh my gosh, if I do all these things, I'm going to be able to handle this genetic risk factor. Another example is even the pathology itself. So, we get hooked on all these pathological changes, but the bottom line is it's not a strict correlation either. We look at alpha-synuclein, we look at amyloid. You think they correlate with behavior? Good luck. No, they don't. So you have these nuns that are studied, right? And they gave up their lovely brains, these sweet nuns. And what did they get studied for over their life? Handwriting, to show that they had these amazing cognitive skills. Hell, half of them had Alzheimer's pathology all over their brain, but they didn't have Alzheimer's disease. So we have a real challenge in front of us, and that is trying to understand what and how much pathology really correlates to behavior, and how much of that changes synaptic function, and how much resilience we have. And so if you believe the idea that lifestyle is creating what? More synaptic integrity. I can afford to lose some synapses. Get rid of them. Who cares? I've got enough reserve or what? Compensatory strategies that make a difference for me in my behavior. And so I think the challenge in front of us is understanding what the limitation of these, plasticity and responsiveness is. What's the limit of that? Probably the most obvious thing is aging itself. So we do know that aging over time changes the characteristics of this plasticity. It doesn't stop it, but it changes the characteristics of that. And aging is a big, so that's a really good field for us to look at as we begin to understand how much can this brain respond to our environmental response and still cause some long-lasting resilience. Honestly, as far as I'm concerned, I feel like there's no day better than today. So I'm always about, yeah, you know, you can still make a difference. Small gains are good gains. Get started now, and I'm perfectly good with that, you know. Although, again, people have tried to look at lifestyle over time with physical activity, and the data is a little confusing. Some people talk about having lifestyle effects when you're very young. Some people talk about lifestyle effects within the last 10 years. Frankly, the problem is being able to retrieve and recall that information. So the lifestyle-related physical activity is a little bit all over the map right now. I think now with what we're capturing with electronic messaging, a lot of these wearable devices, I think it's going to be very interesting to see as we start really getting things more in real time, to see we're going to get some really interesting data out of that type of metric as well. But it's a great time to be in the field because, you know, I'm only talking about exercises as much as I like exercise. I'm not limited to the idea that exercise is the only thing that can be, you know, important here. Obviously, all this wonderful field about mindfulness, right? We talk about sleep. Obviously, sleep is huge, right? All these things that are impacting, you know, this sort of top-down cognitive processing and helping our brain handle, really handle is what we're talking about. That's really what resilience is. Handle disease states. Handle it in a way that we're still functioning at a high enough level. Hell, take away 30% of my cells. I don't care if I can do what? Perform, right? And so in my mind, it's a little bit more of a plausible, believable treatment, right? Are you going to really wait for a stem cell? Or can I tell you if what you're doing right now can make a difference and it's free without side effects? You know, it seems like kind of a silly question. Okay.
Well, so I think at this point we've reached the 2 hours time point. Now we can make this informal. Go drink our booze here.
Yeah.
And
What do you really think, Peter? No, I'm kidding.
no, and so I very much appreciate your coming. I also be very interested in your feedback because we have the opportunity over the course of the year ahead and years ahead to have other programs that are sort of brain health oriented. We have a number of pressing topics of what to do to make brain health better, and we have some ideas of what we're going to be doing here at Ford in the future too. So setting up the dialogue, hearing your feedback, what you'd like to hear the next time we have a community meeting like this is very much welcome, and thank you for coming out.